Okay, well, thank you very much for inviting me to come and speak today. So I'm Dr. Kelly Williams, and I'm a geneticist in the Macquarie University MND Research Centre. And I have worked on trying to uncover the genetic basis underlying motor neuron disease for my entire academic career since I graduated from university. So since today I'm going to be talking about some of the current exciting genetic research that's happening in our field, I thought it would be quite important to actually have a quick background to genetics. So if we start at this slide down the bottom, we have cells. So our entire body is made up of cells. And in every cell of our body, we have 46 chromosomes. So 23 of these chromosomes are inherited from our mother, and 23 of these chromosomes are inherited from our father. And each of these chromosomes is basically made up of very tightly wound strands of DNA. So our DNA code is made up of 3 billion single DNA units, and this is what we call the blueprint of life. So in our DNA code, around 1% are actually made up of what we call genes. And these genes are then translated into proteins, which are the functional units of our cells. And as geneticists, what we are actually specifically looking for is variation in these genes. So we look, when we're reading DNA code, we look for where there's a difference in the DNA code in one individual compared to what is considered a normal individual. So genetic variation, it's quite common. That's why we are tall or short, have blonde hair, brown hair, blue eyes. So as I said, genetic variation is very common, but it's when this genetic variation is actually detrimental, it's causing a genetic disease. It can cause a genetic disease or a risk of genetic disease. So there are two main types of genetic inheritance. So on the left-hand side, we have a more simple inheritance where if you inherit a single gene, you will present with a single trait. So this is where you may inherit a genetic variation from your mother, and you have the same trait as your mother. For example, well, in the example of males, colour blindness. So one gene is leading to one trait. However, we do have complex genetics where we have multiple genes that contribute to a trait, and this is called polygenic. And perhaps the best example of this is actually eye colour, where we can see a different range of eye colours that are coming from genetic variants on lots of different genes. So these two, I guess, modes of genetic inheritance can directly relate to familial and sporadic motor neuron disease cases. So around 10% of motor neuron disease cases, we see a clear genetic inheritance of the disease from one generation to the next. So on the left, this is called a pedigree. Males are indicated by squares, females are indicated by circles, and the individuals that are coloured in black are people that have motor neuron disease. And we can see there is an inheritance of every single of motor neuron disease in every single generation. However, the majority of motor neuron disease cases are sporadic, where we don't see any genetic inheritance and they appear to randomly pop up in the population. So the reason that we consider having motor neuron disease families very important is when you have a complex disease where you don't really particularly understand what's causing the disease or why the disease is being caused, we first go to the families because we know that there is an underlying genetic defect that is causing motor neuron disease in these cases. So at Macquarie University, at the end of 2012, 5 different research teams who all worked on motor neuron disease were all brought to Macquarie University to work alongside the clinic, basically in a research workflow. From the clinic through the different research themes back to the clinic. So I am in the genetics and genomics research theme. So the particular goals of human research, so why are we using these families and sporadic cases to actually, so why are we using these in genetics research? So the first one is for diagnostic tests. So to date, the only proven cause of motor neuron disease are gene mutations that lead to motor neuron death. That's the only proven cause. So if we can identify new genes that cause motor neuron disease, these can directly be added to diagnostic tests. Secondly, we use these gene mutations and we use the gene mutations and the actual genes themselves to try to understand more about the disease. So when we make gene discoveries, we then give this discovery to our model research team, which they try to develop models that will actually mimic the features of motor neuron disease in the laboratory, which is then an avenue to go towards drug testing. Our gene discoveries are also used in our functional biology research teams to, again, try to understand the disease mechanism. So why are these gene mutations actually causing motor neuron disease? And all of this is basically to try to lead to a therapeutic treatment. So this entire research stream, and the reason that we actually do research on motor neuron disease, is to try to find a therapy. So here we have a graph of the current knowledge of causal motor neuron disease genes. So when these genes are mutated, they are causing motor neuron disease in particular families. And we can see the first gene was discovered back in 1993, which makes up around 15% of familial motor neuron disease cases. So no new genes were found until 2009 when Associate Professor Ian Blair and myself and our collaborators in the UK, we identified mutations in a new gene called TARDBP. And since then, there has been many genes actually implicated as causing motor neuron disease. So these gene discoveries are quite important. And so Nim briefly touched upon pathology in motor neuron disease. So this is a histological slide of a motor neuron in post-mortem tissue. And you can see these black clumps that fall within a cell. So these are abnormal proteins. So remember, if you have a gene, it will make a protein. If you have an abnormal gene, so a mutation in your gene, it will actually make an abnormal protein. And what happens is, in motor neurons in the brain, in the cell body of the motor neuron, you get clumps of these abnormal proteins that cannot be gotten rid of out of the cell. And what's interesting is, most of the genes that have been found to cause motor neuron disease, their abnormal proteins are found in these clumps in motor neurons. So when I was putting together this talk, I came across this figure, and I thought it just really well illustrated how important genetic discoveries have been into understanding motor neuron disease. So each of these bubbles are basically publications or research papers that have come out of these genetic discoveries. So they're not necessarily research publications on genetics. They're more likely to be functional biology, so understanding why these gene mutations are causing motor neuron disease, or disease models that are trying to model the disease in the laboratory. So I just thought it was a quite nice just to show the impact that some of these gene discoveries have actually had. So as I mentioned, every new gene that we discover can be immediately added to diagnostic tests for the clinic, and several of the genes that we have discovered at Macquarie are now on this diagnostic screening panel. And also, something very important to come out of gene discoveries is the ability to undergo pre-implantation genetic diagnosis. So this is if you are in a family with a known gene mutation and you're of childbearing age. You can actually choose to have your embryos screened for this gene mutation, and then only embryos that do not carry a gene mutation can then be implanted into the female. And this effectively stops the disease in that family because we know that the gene mutation will not be passed on. So one of the most important aspects of genetic research is actually the patients. So without patients, without DNA samples, we cannot do research on genetics at all. And at Macquarie University, we have set up a MND patient and control sample biobank. So it's a large collection of biological samples from MND patients. So we collect all of these things in the clinic, and our clinical information goes into a patient clinical database where we use the clinical information, so say the site of onset, the age of onset of the patient, the severity of the disease. We then correlate that back to results that we get from our other research themes. We also collect blood and urine to go into our proteomics and our biomarker research themes. We sometimes collect skin biopsies from patients to then culture patient cell lines for our functional biology team to analyse. But our genetics team, we collect 2 tubes of blood, which then get processed in an automated robot to get out DNA and RNA samples, which we use in our research. So the first thing that we do when we get, with every single MND patient that comes through our clinic, we screen for this C9orf72 repeat expansion. And the reason that we screen for this, as what has been mentioned before, is that it is the most common genetic cause of motor neuron disease in both familial and sporadic. So when we screen for this gene mutation, we actually can account for the cause of motor neuron disease in 40% of familial MND cases and around 3% of sporadic cases. The DNA samples from our remaining sporadic cases that don't have this mutation then go into Project Mine, which I'll speak about a bit later. Our familial samples, with these samples, we then undergo DNA sequencing of SOD1, which is the second most common gene mutated in MND. And we end up finding around 15% of these cases have mutations in this gene. So if we combine the known SOD mutations and the known C9orf72 mutation cases, we are currently looking for disease modifiers in these patients, which I will also speak about. And finally, the 40% of patients that are, the 40% of familial MND patients that are negative for our known genes go through our novel MND gene discovery research pipeline. So these are the 3 things that I will speak about for the rest of my talk. So our most recent gene discovery was actually published earlier this year, and it was a massive collaborative effort. So these are all authors that contributed to this research, and they're from all around the world. So I think there's probably 8 or 9 different countries, so clinicians and scientists from all around the world contributing to this study. And we found mutations in a gene called CCNF that is causing both motor neuron disease and frontotemporal dementia. And I'd like to take you through how we actually found this gene and what it means. So we started with a large family that has motor neuron disease, but also has frontotemporal dementia. So individuals in grey have frontotemporal dementia, individuals in black have motor neuron disease. So we started with this large family, and we got DNA samples from all of the younger individuals and some people in the older generation. And what we do is this technique called linkage analysis. So we basically are looking throughout the genome for chromosome regions that have genetic variation, because we all have normal genetic variation. So chromosomal regions throughout the genome that are shared amongst people that have motor neuron disease, but are not in people that do not have motor neuron disease. So you're basically looking for an area, and this is what a result looks like. We're looking for an area where this genomic region is in all people that have motor neuron disease. So we're linking a region to the disease. So we know somewhere on chromosome 16, the mutation that's causing disease in this family lies. So once we have this region, we then performed high-throughput DNA sequencing on 4 individuals that have disease. So this high-throughput sequencing basically looks at the DNA sequence, so it gives us a readout of DNA sequence of all of our 20,000 genes. So we get a readout of that, and then we look at how many variants are in those genes. So amongst 4 people, we end up seeing around 300,000 variants, which is what we would expect, but they're not all bad. So we then look at how many of these variants are actually present in all of the affected, because we assume that if a gene mutation is causing disease, it must be in all the affected people. We then look at ones that are actually variants that are detrimental to the protein, so they are causing an abnormal protein, and ones that are not reported in the normal population. And so we end up, there were only 2 possible gene mutations that could cause disease, and only one of these fell on chromosome 16. And that was a mutation in the CCNF gene. So once we've found it in a family, the next step is to go and ensure that it actually is in all the affected people in the family, so we can see a clear inheritance of the disease. So this is what DNA sequencing looks like, and we can see here that we have this DNA base here is different to the one above, and this is what a mutation looks like. So we were able to see that this mutation is present in all affected people in this family. Next, we go around the world. So we want to actually check that it's not just causing motor neuron disease in this one Australian family. We want to see if it's causing motor neuron disease broadly across the world, and if it's also present in sporadic ALS patients. So we went to our genetics collaborators all around the world. So you'd be pleased to know that we all actually work quite nicely together. We all share our data because we all want to find a treatment for this horrible disease. And so when we went to our collaborators around the world, we did in fact find that there were many mutations in this gene in motor neuron disease and frontotemporal dementia cohorts. So as geneticists, we'd kind of done our job. We'd found a new gene. But how can this actually then enhance our knowledge of motor neuron disease? So our discovery can now be used in diagnostic tests. So remember, these are the goals of our genetics research. Our gene discovery is now being developed into models in the laboratory at Macquarie University. And we have also used this to try to understand the disease mechanisms. So I'll just give one quick slide, because it's quite complicated, even for me, on our functional biology team, how they have used our gene discovery to try to understand disease mechanisms. So, in every cell in our body, not only do we have chromosomes, we also have this system called the ubiquitin proteasome system. And it basically is a whole lot of proteins that form a protein recycling plant. So when there are abnormal proteins in the cell that may be made by a mutation and they're not needed in the cell anymore, they progress. So here we have a protein we don't want. They basically progress through this recycling plant to be broken down to be reused again. And what our functional biology team was able to show is that, so CCNF, the CCNF gene makes a protein called CCNF, and it acts in this recycling plant pathway. And what happens when it's mutated, so in motor neuron disease and frontotemporal dementia, when it's not working properly, it doesn't allow this system to work properly. So you're not getting the recycling or the breakdown of proteins that shouldn't be in the cell. And that's exactly what I showed you here before. So this system, when this system is not working properly, we see these chunks of proteins accumulate in the cell because of this system. So I just wanted to show you how our gene discoveries are actually helping us understand what is underlying motor neuron disease. So now onto Project Mind. So this is where we're using sporadic motor neuron disease cases, so the majority of all cases. And this is a really, really exciting new venture by MND geneticists around the world. So the aim of this is to perform whole genome sequencing of 15,000 sporadic ALS patients and 7,500 controls. So we want to use this massive number of patients to actually look at genetic variants that are contributing to a high risk for motor neuron disease, or an intermediate risk, or a low risk. So because we don't have many large families left that haven't had their gene mutations solved, we're now using a massive amount of sporadic cases to understand the genetics. So this is where we're looking at that polygenic inheritance, so multiple genes causing a trait. So just to kind of give you an idea of what a big job this is, you remember our first genetic slide? So our DNA code is 3 billion units of DNA, and we want to determine the sequence of every single one of these for every single one of those patients. So we use a technique called whole genome sequencing, so determining the DNA sequence of the entire genome. Now, when whole genome sequencing was first completed was back in the 90s with the Human Genome Project, and it cost $3 billion and took 10 years, so basically $1 per unit of DNA. Now, in 2009, a new technology was introduced called next generation sequencing, which basically reduced the cost of genome sequencing dramatically, and also the time to do it. Now, if we fast forward to 2016, we're currently genome sequencing 600 Australian sporadic motor neuron disease cases, and the sequencing provider that we use that's in Sydney, it cost $1,750 per genome. It only takes a few hours to sequence all 3 billion base pairs, and they can do multiple individuals at a time. So they're actually getting through 70 a day. So technically they should be finished in 2 weeks, but we'll see about that. So many of you would remember the Ice Bucket Challenge, and it raised a massive amount of awareness for motor neuron disease, but a lot of the money that was raised came to a lot of the money that was raised in Australia came to a group of 16 researchers from all around the country, or 16 researchers and clinicians from all around the country, to basically set up or establish an infrastructure for collecting sporadic DNA samples and associated clinical information. So we have this amazing cohort of patients that we can actually do some really, really good genetic studies on. So it's led by Associate Professor Ian Blair from Macquarie University and Professor Naomi Ray up at the University of Queensland. And it involves more New South Wales clinicians and researchers. So we have Professor Dominic Rowe, myself, Professor Matthew Kiernan, Professor Garth Nicholson, Professor Steve Vucic, and Professor Roger Pamphlet, who'll be speaking later. So a lot of the top geneticists and several of the clinicians in New South Wales are working directly on this project. So some results actually came out this month from Project Mine. So from the first samples that were sequenced, so a lot have actually been done in Europe as well. So 2 research papers came out this month, basically identifying new genes that are associated with motor neuron disease. So NEC1 and C21ORF2. So this top research paper here, NEC1, it was actually discovered using 1,000 familial ALS cases. So they weren't in large families, they were all single familial ALS cases. It included probably about 100 from Australia. And they looked at all of their genomes and found an overrepresentation of detrimental genetic variants in this gene. They then went to look at the Project Mine cohort, so several thousand sporadic individuals, and found that they could replicate this. So they actually found in sporadic individuals as well, there was an overrepresentation of these detrimental variants in the NEC1 gene. This paper here was using purely sporadic ALS cases. So C21ORF2 basically means chromosome 21, protein number 2. So we actually have no idea what this protein does yet, but it does open many, many more avenues of research. So I think it was 20,000 sporadic ALS cases were actually used in this study, and they looked to find genomic regions where there were clusters of detrimental gene variants. And they found several regions that we already knew about, including the C9orf72, and then this was the new one that actually came out of this research. So this is using, tens of thousands of patients to actually get new gene variants that are causing or that are associated with motor neuron disease. And remember, this is our major aim as geneticists, and now we give these discoveries to the rest of our research pipeline to try to understand why these are causing motor neuron disease. Okay, and finally, I will talk about the research looking for disease modifiers in familial MND, and this is the research that I currently work on is very close to my heart, and I think a very important part of our research program at Macquarie. So here is a graph showing the breakdown of the mutations that cause familial motor neuron disease in Australia. And we can see that more than 50% of cases are by mutations in just these two genes. So we still have this big chunk of unknown. We have rare genes here, and we have more than 50% in these two genes. So you may remember back in my early slides, we talked about inheritance, where you can get a single gene variant leading to a single trait. And that's generally what we call a genotype-phenotype correlation. So this is your genotype, so your gene code for a specific variant, and how you're presenting with disease. And we don't actually see this all the time in these cases. So these individuals, they have the same gene mutation, but they don't always clinically present exactly the same. So we have a family, one of our families looks like this, where we have individuals that have motor neuron disease, so pure classic motor neuron disease, individuals that have frontotemporal dementia, and individuals that have both. So this is just in the one family, they all have a C9orf72 mutation. So we call this comorbidity. In our cohort of patients that have mutations in these genes, so C9orf72, SOD1, FUS, TARDBP, ubiquitin-2, we see varying lengths of disease duration. So these ones in red are rapidly progressive individuals, and these ones here in yellow are slowly progressive individuals. And this is probably the most exciting aspect of this study, is the fact that we do have a difference in disease duration. So there are factors that are causing these individuals that have the same gene mutation. There's factors causing these people to actually live longer, so they have a more slowly progressive disease path. And if we can actually find out what these specific factors are that are causing these individuals to live longer, and if they're treatable by drugs, it gives us an opportunity to actually develop therapies to prolong the disease course. So it's not a rapid course. So we would turn these red people into yellow people, and that is the major aim of this part of the study. But in ALS, we also see a massive variation in age of onset. So I know Nim did mention the average age of what we see patients present in the clinic. But in our clinic at Macquarie, we have individuals in their 20s that have motor neuron disease, and also individuals that come in first presenting in their 90s. So we have this, again, we have this large discrepancy in the clinical presentation of these patients. So in order to actually try to understand why we're seeing this variation, we're looking at 3 different genomic levels where we might find these modifying factors. So I've mentioned DNA sequence variation. So these individuals, yes, they have identical gene mutations, but they have lots of other variation in their genome. So we want to see if any of this other variation is actually causing them to have early onset or late onset or rapidly progressive or slowly progressive motor neuron disease. We also are looking at RNA. So DNA is translated into a protein, which is kind of over here. I didn't have enough room to put everything on my slide. But the intermediate step is RNA. And that, from RNA, we can determine the level that we will see of a protein, because it's that intermediate stage. So if we see lots of RNA of a certain gene, we know there should be lots of protein of that gene made as well. So we can, again, we want to have a look at RNA expression levels in all of these patients to see if there is a difference in their expression levels of particular genes, which could also help us understand the disease better. Probably the most complicated thing when I normally try to explain this in talks is DNA methylation. So DNA methylation is actually found upstream of a gene. And it's a methyl group, so it's a carbon and 3 hydrogens, which is physically attached to the DNA and stops the DNA becoming RNA and becoming a protein. It physically prevents anything from turning DNA into proteins. So DNA methylation changes all throughout our life. So even identical twins have different methylation profiles. So we are looking at these 3 different genomic levels to see if we can find clusters of these genomic levels in certain subsets of patients. So what we're doing, we take all of these patients. So from the collections of Professor Garth Nicholson and Professor Dominic Rowe, we have, Garth Nicholson in particular has been collecting DNA samples for 30 years now. So we have more than 200 patients that have mutations in these 2 genes. And we then, as I mentioned, we're going to be looking at their DNA, their RNA, and their methylated DNA. And this is what we hope to see. So we hope to see, so for example, this could be a specific variant in a specific gene. And we can see that it's present in, maybe 4 out of 100 people here, but then it's present in 7 out of 20. So we can see that this variant is clustering in late onset people. And what we hope to use this for, we can't change genomes yet. So what we've got, we're kind of stuck with. But we can use this as a prognostic tool. So if someone came into the clinic and we said, oh, well, we've sequenced your genome and that we see that you've got more of these variants that generally cluster with later onset people, if it's a hereditary case, we could say, we believe that you may not get disease until later in your life. So that's what we're hoping to do with the genotypes. Now with RNA, with gene expression and methylation, because these fluctuate throughout our lifetime, they are actually modifiable by drugs. And that is where I think we have this study. Obviously it's in its infancy stage, but there's real promise that we can identify modifying factors that can undergo therapeutic treatment. So finally, just wanted to recap some outcomes of gene discoveries that we've been involved in. So when we identify gene mutations, it allows us the opportunity to mimic the disease or the features of the disease in the laboratory, which will then allow us to potentially test drugs. But we have done, we have done this. We haven't done drug testing in the labs yet, but we have developed models of the disease. It also gives us a better understanding of motor neuron disease. So we know gene mutations are what can cause motor neuron disease, but we don't know why. So by using our gene discoveries, the functional biology research teams are learning more and more about the underlying disease mechanisms. And finally, for those that have a family history of motor neuron disease, so the familial cases, the 10% of cases, we can offer DNA testing which will give an answer in 60% of those cases, which when I started my career, this was 10 to 15%. So a lot has happened in the past 9 years. So if we find a gene, at-risk family members, so they could be siblings of an affected individual or children of an affected individual, can undergo genetic counselling to then get tested to see if they also carry this gene mutation, which can then lead to them having, I guess, conversations and options about reproduction. And the main one of this is IVF and pre-implantation genetic diagnosis. So if you know the gene mutation in your family, you can actually prevent passing it on to the next generation by undergoing these procedures. Now there is more information on this on the MND Australia website if there are individuals here that do have the hereditary form of the disease. Other than that, I'm more than happy to take questions and to speak to anyone at lunchtime who might be shy about asking questions. So thank you.